Question 16. Question 16 asks that study the given figure of nucleosome, that is figure of nucleosome, and select the option that correctly identify the part A, B and C. So if we see the diagram, in diagram A is representing histone octamer, B is representing DNA and C is representing H1 protein. So if we match according to this, then in a sequence of ABC, any option is not matching, okay, but you should remember that A is histone octamer, D is, according to option, if we see there is a typing mistake in option C, they have written histone, so you should histone octamer, then DNA and then H1, so that become the correct, H1 histone, that become the correct option. So C can be most possible answer, but it is not matching. Modify the option C in the form of histone octamer, DNA and H1. Question 17. Question 17 asks that it is absent in RNA. Now in option they have written option A is pentose sugar. Yes, RNA has a ribose sugar, and ribose sugar has a 5 carbon, so it is a pentose sugar, obvious. That is correct. B, uracil. Yes, uracil is a nitrogenous base which is present in place of thymine, which is found in DNA in place of thymine, DNA, and RNA. Uracil is present. C, guanine. Yes, guanine is present. And D, adenine. Adenine is also present in RNA. Now actually it is not, C option is not guanine, it is a guanine, and there is a guanine, not a guanine, so C is not present in RNA, so C is the correct answer. Question 18. Question 18 asks that which of the following is required as inducer? Required as inducer for the expression of leucoperon. Now inducer bind with repressor and inhibit binding of repressor with operator. Now in case of leucoperon, lactose and allolactose act as an inducer. So according to this, lactose is given in option is A, so option A is the correct answer. Question 19. Question 19 asks that choose the incorrect sentence for RNA. Incorrect sentence for RNA. Now if we talk about incorrect sentence of RNA, A is given as RNA is useful for protein synthesis. Yes, RNA is useful for protein synthesis. Option B, tRNA brings amino acid and reads the genetic code. Yes, tRNA brings amino acid and reads genetic code. Option C is given as RNA contains 2 polynucleotides. Actually, RNA have single polynucleotide chain, so it is incorrect. And option D, during translation rRNA act as a catalyst, so it is correct. So C is the incorrect sentence for RNA. Question 20. Question 20 asks that how many genetic code proline has? So if we talk about a proline is an amino acid. It has 4 genetic code. These 4 genetic code are CCU, CCC, CCA and CCG. So these 4 are there. So C option given as 4, that is the correct answer. Question 21. Question 21 asks that Okazaki fragment were formed. So option A. So first we should understand what is Okazaki fragment. When there is a replication, there is a synthesis of one continuous strand, that is known as lagging strand, and there is a discontinuous synthesis on the another strand, which is known as leading strand, and that is lagging strand. Now on lagging strand, these fragments of DNA and RNA, these are known as Okazaki fragments, as this was discovered by Reiji Okazaki. So if we go with the option A is given as on the continuous strand at the time of DNA replication. So not on the continuous, on the discontinuous strand. So it is incorrect. Option B is given as on the discontinuous strand at the time of DNA replication. Yes. Option C is given as at the time of transcription. No. And option D is given as from the introns at the time of transcription. No, not from the introns, it is from the particular DNA sequence. Not only introns, exons also be there. So B is the correct answer. Question 22. Question 22 asks that the 3' to 5' phosphodiester linkage inside a polynucleotide chain serves to join. So 3' to 5' phosphodiester linkage. Now when there is a 2 nucleotide that have a sugar, at first carbon there is a nitrogenous base, at third carbon that is a OH, and at fifth carbon there is a phosphate. Now when there is another nucleotide, at first carbon nitrogenous base, this is the sugar, and at fifth carbon there is a phosphate. Now the bond which is formed here between this OH and phosphate group, that is known as phosphodiester bond. So in this there is a joining of 2 nucleotides. So phosphodiester bond is used to serve to join one nucleotide with another nucleotide. So C is the correct answer. Next is question 23. Question 23 asks that by using diagram, select the correct option for X and Y. Correct option. So now in this, first option given is here X, means X is replication. Then here transcription is written, mRNA, translation, and Y. Now this Y represents protein, and this is a central dogma diagram. So replication is X and protein is Y. So A is the correct answer. Question 24. Question 24 asks that DNA is the genetic material because DNA is the genetic material because option A, DNA is the chemically and structurally stable material. Yes, it is chemically and structurally stable. Option B, it having a characteristic of self-replication. Yes, it can replicate by its own. C option is given as expressed in the form of Mendelian character? Yes, it is expressed in the form of Mendelian character, and all the three are so given, so all are true is the fourth option, that is option D. So D is the correct answer. Question 25. Now question 25 asks that in capping, blank is added at 5' end of hnRNA. Now there is a 5' capping, which is the first step of post transcriptional modification, and this in 5' capping, when hnRNA is there, which is also known as primary transcript, at the 5' end there is addition of a cap of 7-methyl guanosine triphosphate by enzyme guanyl transferase. So 7-methyl guanosine triphosphate, that is the correct answer, that is option A. Question 26. Question 26 asks that which of the following is correct for mRNA in eukaryotes. Now if we talk about option A is asking that exon appear in mature RNA. Exon appear in mature RNA, but intron not appear and are absent or not appear in mature RNA, then it is correct. Option B, exon and intron do not appear in mature RNA. Exon appear, intron do not appear, so it is a incorrect statement. C is given as intron appear but exon do not appear. Actually exon appear, intron do not appear, so it is also incorrect. And the exon and intron both appear in mature RNA, so it is also incorrect. So A is the correct answer, that during splicing, during splicing, introns are removed to form mature RNA. Next is question 27. Now question 27 asks that which of the following group of codon is a stop codon. So stop codon or termination codon are the codon for which that the stop codon are the codon which do not code for any amino acid, and this stop codon are UAA, UAG and UGA, UAG and UGA, that is option B. So B is the correct answer. Question 28. Question 28 asks that which enzyme will be produced in a cell if there is a nonsense mutation in lac Y gene. Now this is the question of lacoperon. Now you know that the diagram of lacoperon is somewhat like this, where you will find a I gene, which is a regulator gene, then promoter gene, operator gene, then lac Z, lac Y and lac A. Now the question is asking that there is a nonsense mutation in a lac Y gene. Now lac Y gene, there is a nonsense mutation. Nonsense mutation are the mutation which convert codon into stop codon. So if it convert into the stop codon, then termination of polypeptide will occur. Means it will not cross, RNA polymerase will not cross beyond leg Z and it will terminate here. So there is a no mRNA or polypeptide for the leg Y and leg A gene. But leg Z gene express, and leg Z gene will form a beta galactosidase enzyme. So option C is the correct answer. Question 29. Question 29 asks that which of the following is not correct option for tRNA? Not correct option for tRNA. Now tRNA is a transfer RNA and it is also known as adapter molecule. It helps to take amino acid during a process of translation. So if we go with the option A is given as it has an anticodon. Now we have to select a not correct. It has an anticodon. Yes, it has anticodon at the anticodon loop. Option B, it has an amino acid acceptor site. Yes, at a 3' end that has amino acid acceptor site. Option C, tRNA are not specific for each amino acid. Actually, tRNA are specific for each amino acid because 61 tRNA are available for 61 codon. So it is incorrect. And D is saying that there are no tRNA for stop codon. There is no tRNA for stop codon. So it is a correct statement. So incorrect is option C. Question 30. Question 30 asks that which of the following is responsible for transcription of tRNA and snRNA. Now actually if we talk about the RNA polymerase, then there are three types of RNA polymerase in eukaryote, that are RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Now RNA polymerase 2 code for hnRNA and mRNA, whereas RNA polymerase 1 code for 28S rRNA, 5S rRNA, but 5.8S rRNA, tRNA, snRNA, scRNA, that all are synthesized by RNA polymerase 2. Now the question is regarding tRNA and snRNA. So the option C, that is a RNA polymerase 3, that is option C is the correct answer.